Bom dia, pessoal. Estamos aqui mais uma vez, iniciando agora a segunda semana do nosso curso. É um prazer novamente estar aqui com vocês. Lembro a vocês, com relação à nossa dinâmica aqui do curso, que todos os dias nós temos que assinar a lista de presença. A lista de presença consta num link, né, abaixo aí dos outros vídeos, de todos os vídeos de cada aula, então vai ter uma lista de presença específica e que está localizada aí, então vocês já podem entrar a qualquer momento e assinar a lista de presença. É extremamente importante que assinem, porque a dinâmica do curso requer que no final nós contabilizemos a presença para o oferecimento dos certificados. Então, espero que vocês estejam todos bem. Essa semana novamente vai ser um pouco puxada, porque nós temos uma série de palestras, palestrantes muito bons. E hoje nós teremos mais um palestrante excelente, referência internacional nos estudos de dinoflagelados. Nós teremos conosco aqui, então, o professor António Calado. O professor Antônio Calado, ele é atualmente professor auxiliar no Departamento de Biologia lá da Universidade de Aveiro, de Portugal. E ele é biólogo e ele tem doutorado na Universidade de Copenhague. E, além dos dinoflagelados, ele também se interessa, é claro, pelas diversas temáticas das algas. Mas os dinoflagelados têm sido um objeto de estudo mais intenso do professor. Inclusive trabalhando com taxonomia, com filogenia, né? Então, eu gostaria de convidar aqui para vocês o professor Antônio Calado. Olá, professor.
Bom dia.
Bom dia. O professor é português, mas ele preferiu dar a palestra dele em inglês, diante do fato de que pode facilitar melhor a compreensão de outras pessoas, de outros participantes que não do Brasil, né? Então, ele optou por isso, mas também fiquem à vontade para fazer depois as perguntas em português, que ele vai compreender com maior facilidade também. Então, deixo a palavra para ti, professor.
Okay. Muito obrigado. So I will switch to English and I will start the presentation. I guess that you are now seeing a shared screen. Is that right?
Yep.
It looks like a landscape somewhere in North America. I will start the presentation with the first part. Now I was left pretty much with great liberty by the organizer. And I know that this is within the framework of continental algae. So you are primarily interested in algae that occur mainly in freshwater. However, the group that I will be presenting, the dinophytes, the dinoflagelates, if you want, are a group that occurs both in freshwater and marine water and all kinds of brackish water, so it's actually very widespread. And some of the general concepts will actually be more easily illustrated by some marine examples. So there will be a change or a set of examples that will apply perhaps only to the marine, or will have some freshwater examples and so forth. So the first part of my presentation, I hope this fits what you are expecting or what you need, is an overview of the group of the dinoflagelates. As I said, a bit more focused on freshwater things, but with some indications about at least some of the main features, even if they occur mostly or only in some cases in the marine species. So I hope this is something close to what you have been expecting. Let's see. So the group is, if you want to call it, with formal name for a class. It could be Dinoflagelados. Some people raise it to the status of a phylum, and that would be Dinophyta, and it's normally placed phylogenetically close to a couple of other groups, making the assemblage, which is called alveolate. We'll go into that a little bit further on. And you can see some images here, some simple drawings that show a few of the general morphologies that you will find in the group. So when we just briefly consider dinoflagellates, we may say that it's almost entirely made up of unicellular organisms. The vast majority of them are single-celled, so that would be a very general situation for them. There are photosynthetic species, so some of them will have chloroplasts, and that's of course a requirement if we want to call them algae. That's why they are considered in this context. But others do not have chloroplasts at all and rely on different modes of heterotrophic nutrition. And there are quite a number of them that combine the presence of chloroplasts and the capacity to feed on organic material. Many of them even feeding on particulate material, which means that they can uptake organic particles, and these are very often either bacteria or other eukaryotic organisms. So this mixotrophic strategy is used by many algae and certainly by many dinoflagellates. There are also some parasitic forms, some of them actually of some practical importance, and some of the forms are involved in symbiosis, and here again dinoflagellates become very prominent because some of these symbioses are absolutely essential for some of the world's most diverse habitats. I'm talking, for instance, about the corals, which are the most diverse habitats in the marine environment and which are very much dependent on the symbiosis that the corals, the animals, engage with mostly with the dinoflagellates that give them the photosynthetic capability. Now, the image on the right-hand side of your screen is the image that shows slightly artistic drawings, mostly of a group of, I would say most of the species in there would be marine dinoflagellates. This plate is authored by this person whom you probably know. It was the person that first coined the term protista to separate this group of eukaryotic organisms from the traditional animals and plants. So it was quite a revolution. And so it was Ernst Haeckel, and he produced this very famous and very beautiful set of illustrations of virtually everything in the living world, from animals to plants to different kinds of protists, in the general book that is, with the name Kunstformen der Natur, which means art forms in nature. So this is where those images come from. But I have here some overview of forms that are found in freshwater. So in freshwater dinoflagellates, you have certainly some diversity, quite a lot of diversity, but not as much as when you consider some of the very spectacular forms in the marine environment. In any case, we can see here some of the main themes that will occur in dinoflagellates. And one of them can be seen, for instance, in cells like this one or this one, which is the presence of a furrow that goes around the cell, sort of dividing the cell into two halves. An upper one, which we will refer to usually as the epicone, even if it's not conical. So it's just the upper part above this transverse furrow, and the other one will be, of course, the hypocone, the one that is behind. And behind here means the one that is behind when the cell swims. The cells mostly have two flagella in the vegetative stage. The flagella can be seen, for instance, this one here is drawn inside the transverse furrow going around the cell, and then a second one is going along what is seen here clearly as a longitudinal furrow, and then emerges on the back of the cell. Again, I say this is the back of the cell because when the cell swims, it will swim to the side that is here turned upward. So we have a natural orientation of the cell according to its direction of movement. And if we consider the insertion of the flagella as a reference point and call this area the ventral area, then we actually have a possibility to refer to all parts of the cell in a way that is analogous to what we would do with an animal like ourselves. So this will be the ventral part. Of course, the opposite part, which is shown here, for instance, in this cell, would be the dorsal part. This is the anterior end, that would be the posterior end. And of course, this is right on the cell, and this is left on the cell, which when the cell is ventral to us is, of course, on the other side. That would be the left part of the cell, and this would be the right part of the cell. So these are the sort of the cardinal points of a dinoflagellate. And you can see some of them develop some particular shapes, and some have these projections like this one here, which is called serraceum because of the horn-like projections that it shows. Some of the species, especially in the marine environment, depart from this morphology in not having furrows. They don't have a longitudinal furrow, they don't have a transverse furrow. By the way, transverse furrow, we normally refer to that as cingulum because it resembles a belt around the waist, whereas the other one is often referred to as sulcus. So some of the species do not have cingulum sulcus. Here's an example of that. This is actually an example from freshwater, but there are only 2 species of this group known from freshwater. This kind that does not have furrows and has the flagella coming out of the anterior end. We will see a little bit more of that later on. So it is quite common in the marine environment. It is very rare in the freshwater environment, but it exists also there. So I have represented it here. And then there's a group of species which normally will spend most of their lifetime in what is called a coccoid stage, surrounded by a wall and without flagella. So these will produce only the flagellate stages, like these 2 cells that we see inside here, just at the moment where they will divide and propagate. So dispersion will be the primary function of these cells, sometimes also sexual reproduction, in which the cells will behave as gametes and will fuse together. So this is one of the kinds that we see in the freshwater of these so-called coccoid dinoflagellates. This is another kind, if you want, you can call it coccoid, many people do, although in this case there's this abundant mucilage around the cells that will form these groups, and so I guess that some of you would actually prefer to call it palmalloid instead of a coccoid. So these 2 groups also exist in freshwater, although undoubtedly the groups with this general morphology are much more common than we find. So if we look at the general dinoflagellate cell of the most common type, we can see something like this. The cell has a furrow, and inside the furrow, or along the transverse furrow, we have a very special kind of flagellum that is bent in an helical way, and in some areas we see that there is a sort of a membrane like a flat area that is like the stretched cell membrane, and it is stretched between the microtubular system that supports and moves the flagellum and a fibrous rod that stays closer to the cell and so does not accompany these turns of the microtubular part. The microtubular part is, of course, the axoneme, and the distance between the turns of the axoneme and the inner fibrous rod will make the flagellum look a little bit like a ribbon instead of a whip, which is what we normally see in flagella, and actually it is what we normally see in this longitudinal flagellum that goes to the back of the cell. So they are quite different, and they are quite characteristic of the group of the dinoflagellates. Now here we see several features. I don't want to go through all of them now, but we can notice this very large nucleus, which here is elongated and lies at the back of the cell. And inside the nucleus, we see these very large chromosomes that are also usually very numerous, very large. So there's a lot of DNA in here, but we can see that the organization is significantly different from the organization we are used to see in eukaryotic organisms. So that's another one of the peculiarities of the dinoflagellates. You'll see dinoflagellates are very rich in peculiarities. In fact, it's one of the most diverse group in terms of different strategies and things that are really different from what other organisms, what other algae are doing. Now here we see, of course, these areas which represent chloroplast lobes. So this is one of the photosynthetic forms, and it has this sort of a brownish yellow color, which is the most common color in the chloroplasts dinoflagellates. When the dinoflagellates, when the chloroplasts have this type of color, they have a particular type of pigments, and they have a particular type of structure. We can see the structure represented in this part of a chloroplast lobe, which is taken from a freshwater dinoflagellate. And what you can see here is, if you look at this area, of course we have here the membranes that are close to the surface, but when you look at the chloroplast, you can find here three membranes surrounding the chloroplast. So not the two membranes that we find in the chloroplasts of higher plants and in the green algae, but three membranes. And what we see here is the thylakoids, and you can also clearly count three thylakoids together in most of the length of these thylakoid lamellae. So they are very clear, three here, then of course one of them stops there, and there's a little bit with only two, but then another one joins here, so there's three again. So most of the length of the thylakoid lamella has three thylakoids attached together. So this is the general organization, fine structural organization of chloroplast of the main type, the type that you find in most of the photosynthetic dinoflagellates. And this type of chloroplast will typically have this type of pigments. The chlorophylls that we see here are the chlorophylls A and C, or at least one, actually two in some species, types of chlorophyll C. There's of course beta carotene. That's ubiquitous carotenoid in the chloroplast systems. And then there's a number of xanthophylls. Some of them are not exclusive of dinoflagellates. You'll find them in other groups. But this one here that is underlined is the most typical xanthophyll that you will find in this type of dinoflagellates. meaning the dinoflagellates with this type of chloroplast that we have just seen. And this, in fact, this relationship between this type of chloroplast and the presence of this xanthophyll peridinin is so strict, so intense, that we normally call these chloroplasts as the peridinin containing chloroplasts, so the peridinin type of chloroplast. But you can have variations, like almost everything in the groups of dinoflagellates. Just to remind you, chlorophyll C is this type of chlorophyll. You remember that chlorophyll A and chlorophyll B, they both have a long chain of carbons that are attached to one of the four pyrrolic rings. So this is very characteristic for these two chlorophylls, which are the ones that you find, for instance, in the land plants. So this type of chlorophyll, chlorophyll C, does not have that, and it is at least speculated that it is evolved primarily to serve in aquatic organisms. So there is some kind of advantage taken from the loss of that chain. There are several of them, at least 3 of them, which are represented here. They differ in the substitutions that you get here in these 2 points. They are marked R1 and R2, and you can read down here which are the modifications that make them chlorophyll C1 and C2 and C3. There probably are others more that have been described later, but these are the main types. And this here is the xanthophyll that is called peridinin. So that's the structure of it. So this biochemical characterization of algae, that's probably something that you have grown used to. It is usually what you find in the books when you want to characterize a group of algae. Normally, one of the first things that is told about them is what their chloroplasts have in their photosynthetic system, but also this biochemical characterization usually includes the main food reserves that the organism develops. And in the case of dinoflagellates, it's actually starch. Starch, as you know, is the alpha-1,4-glucan, similar to what the land plants produce. So this is the one of the few groups of algae that will, apart from the green algae, of course, that will produce this type of polysaccharide. But here, unlike what you see in the green algae and unlike what you see in the land plants, the starch is formed outside the chloroplast. It's in the cytoplasm. And you find in many dinoflagellates oil droplets which can actually accumulate to significant amounts in some cases. This is what we can see in this cell. Let's see if it grows a little bit. So you actually see that in the upper part of the cell, in the epicone, you see a lot of these oil droplets, and this very divided area, whitish grains here in the antapical area are actually starch grains, which in this case accumulated mostly here in the back of the cell. So that's Peridiniopsis borgii. That's actually a freshwater species, and it shows this sort of separation between the two types of food reserves. So when we look at another cell in a thin section transmission electron microscope, this is actually the whole cell, and it was sectioned through the area where the flagellabases can be found. Some of the roots are here. This is actually one of the types of eye spot. I won't be talking much about eye spots, but just to say that they are also much more diverse in dinoflagellates than you usually find in all other groups of algae. You can see some oil droplets up here. You can see this structure, which will become significant a bit later on. That is here stretched. But what you can see here very clearly is all around the cell, the cell is sufficiently small for us to have a view of the whole cell and still have sufficient magnification to look at membranes. So you can see that the outer part of the cell is very well preserved in this fixation. There's this continuous membrane all around that continues on this produced part of this upper part of the cell. But you can see that whereas here the membrane is alone covering the cytoplasm on this projected part, here you can see that there is something underneath. And what you see underneath here is flat vesicles that are abutting one another in these points there. So you have a layer of flat vesicles very closely pressed to the inside of the cytoplasmic membrane. And this is a very characteristic arrangement, which in the dinoflagellates is called the amphiesma. Here we have a schematic drawing, rough drawing, that was originally published by Max Taylor in 1980, where you see these vesicles drawn. I think it's a bit rough the way he drew them, because you don't see them touching one another here, but they actually touch like you just seen in the photograph. And here you can see that he drew them as a sort of a general flat vesicles with a line inside. And this is because sometimes you have these flat vesicles looking just as if they were empty in the electron microscope, like the ones we just saw. Sometimes you see material in there. This material is generally the polysaccharidic in nature, so something very close to cellulose. And when it is present, then you end up having a surface that sometimes you can see with some sculpturing all around the cell. Usually we call this arrangement with these plates of material and this sculpturing, we call it this armor of the cell, and then we say we have an armored dinoflagellate. If there is no thick material inside of these vesicles, then we say that the cell is naked and we have a naked dinoflagellate. Now, the thing interesting about this type of cover is that although it is quite unusual, it is not unique in the sense that we find this type of arrangement similar to the amphiesma in other groups of organisms, and it is considered to be a unifying character in the group that we call the alveolata. And what are these two groups that contain also these alveoli? Well, one of them is the group of the ciliates. Ciliates have never been considered algae. They're not in the algal books, which sometimes you may think it's a bit unfair, because some of the ciliates actually harbor symbionts that can be photosynthetic, and some of them actually have reduced the symbionts to just chloroplasts. So it means that some of the ciliates will actually have chloroplasts that have been originated from a symbiosis, but then so have all the other chloroplasts of all the other groups. So if there were not so many ciliates that are strictly heterotrophic, then maybe we would have the ciliates in the algal books. In any case, phylogeny confirms that these are close relatives of the dinoflagellates. But even closer relatives, and also with this type of surface, are the group that is called Apicomplexa, or in some of the books you can read about them under the name Sporozoa. And the Sporozoa are perhaps a bit more surprising as relatives of dinoflagellates, because they are, at least nowadays, entirely a group that lives parasitically in the blood of vertebrates. So how does a group that is completely parasitic in the blood of vertebrates relate to this? Well, phylogenetic analysis tell us that they do, and this similarity in the surface is a sort of a clue that, in fact, they are closer than they seem at first sight to both the ciliates and the dinoflagellates. Also, if we consider that it has been discovered, actually it was communicated the first time, I think in 1996, that the apicomplexa, which include organisms that are very well known, like Plasmodium, the causative agent of malaria, that they actually have an organelle that is the remnant of a chloroplast. So it means their ancestors actually had a chloroplast. And the organelle is still there. It does not function in photosynthesis, but it does have a function in lipid metabolism that is essential for the organism. So they can't get rid of it. They have this remnant of a chloroplast. So this idea of algae and what ended up being an algae and what did not end up being an algae is very much the result of what we could see in the organisms today, but it does not reflect in an accurate way their evolutionary origins. So where are the alveolates? I've used this perhaps a bit outdated phylogeny, but it is simple. So we can use this to see where they are. So the alveolates are right here on the top right side. And here we see the dinoflagellates, they're the apicomplexans, and here are the ciliates. So the three groups that contain these flat vesicles underneath the plasmalemma, which justifies this name, alveolates. And close to them, the closest neighbors are on the one side here, the ones called straminopiles, that include the diatoms, that include the brown algae, but also include the Oomycetes group that is normally found in fungi books. And on the other hand, a group that is mostly made of what you would think of as protozoa or heterotrophic protists, including foraminiferans, for instance, including the Cercomonads. Again, this is a matter of perception that they never ended up in the algal books. Actually, some of the Cercomonads did end up in the algal books. If you think of groups like the one that is normally called as chlorarachnophytes, with the chlorarachnin being an enigmatic organism for a very long time, and people didn't know where to put it. It had chlorophyll A and B in its chloroplasts, but it made this rhizoids, this completely amoeboid shape, so it didn't really fit anywhere. And that's because it was not, it did not belong anywhere else. It belonged in a group that seemed to have no affinities with the algae. Even the foramens, the foraminifers, they are unicellular amoeboid organisms that form these very intricate shells, calcareous shells. And some of the foraminifers actually have symbionts, and some actually have symbionts that have been reduced to chloroplasts. So they also have acquired chloroplasts as other organisms have. It's just a matter of being few of them with chloroplasts in a very diverse group that has probably not been enough for people to put them in the algal books. But you can see what the modern phylogeny tells about these things. Another version of this actually has the three groups put together under this name, SAR, which is, of course, based on the initials of the three groups that we saw there, the straminopiles, the alveolates, and the rhizaria. Well, anyway, this is has been relatively stable, this relationship, so that's where dinoflagellates usually appear in the eukaryotic tree of life. Now, I said that the pigments and also the structure of the chloroplast may vary, and they do in dinoflagellates probably more than we see variation in other groups of organisms. So whereas we normally will have this in this peridinian type of chloroplasts. We have chlorophylls A and C, C2, sometimes C3. There are some dinoflagellates, very few, but a few, that will show chlorophylls A and B. Where does this come from? Then in some of them, some of those that we actually find in freshwater, you can see that there are phycobilins. Phycobilins are these accessory pigments that we find in cyanobacteria. And we find them also in the red algae, but normally in other groups, in most other groups, they're gone, but not in all of them. So phycobilins find their way into some dinoflagellates. You can also have other xanthophylls, including, in some cases, fucoxanthin. Fucoxanthin is what you see as the causative of the brown color in the brown algae. So clearly, if you put together fucoxanthin in a dinoflagellate with some other characteristics, then there's a strong suggestion that you have a recent acquisition of a brown alga that is acting as a chloroplast. And this is indeed what is happening when we look at this image with mostly with photographs of eukaryotic groups that contain photosynthetic organisms. For most of them, of course, we have the animals here. But even in the animals, we have symbionts. We know that. We have cases where some organisms can actually retain chloroplasts. So the basis of the process is also here, but most of these organisms are non-photosynthetic. But most of the others that are shown in this board are photosynthetic. And we can see down here what the path of acquisition of the chloroplast was. We have those that acquired chloroplast directly from the uptake and stabilization of cyanobacterium, free-living cyanobacterium that became installed and coordinated its activity with the host nucleus and then became a chloroplast. This is the primary branch that then divided into three branches: the green algae that ultimately gave rise to the land plants, the red algae, and a relatively small group, the glaucophytes, that have retained in their chloroplasts much of the characteristics that we see in cyanobacteria, so much so that the name given to the chloroplasts of the glaucophytes is usually cyanelles. So this is the basis of the acquisition of chloroplasts, and then from here other groups actually got their own chloroplasts. So we have examples on this side from groups that got the chloroplasts from green algae, and on this side groups that got their chloroplasts from red algae. All this is pretty much general, and we see this happening in other groups of organisms. What is special about dinoflagellates is that yes, they got their chloroplast, their peritonean chloroplast, apparently from a red alga, but in several cases, what we see is a replacement of this original chloroplast with something else. And if we look a little closer to this side, the side where we have the dinoflagellates, we can see this one represents a typical peritonean chloroplast containing dinoflagellate, but this one is different. Its color is much greener. So the chloroplast here, the original chloroplast with peritonean, was lost and replaced with the chloroplast which has origin in a green alga. Here we see a completely different color. Very often in the microscope, they look bluish. We used to call them, when I was a student, they were the blue gymnodinium. Now we know that the blue comes from a different symbiont. It's actually a cryptophyte, and the cryptophytes are the organisms, the eukaryotic organisms that got their chloroplast from a red alga, when they did not get rid of the phycobilin pigments. So they still have some of the pigments that make the blue-green algae blue, and that's where the bluish color is coming from in this case. And it's actually a very interesting case where this symbiosis is actually incomplete in the sense that the cell cannot keep their chloroplasts working indefinitely, will eventually lose them, and has to eat the scriptors again, to get chloroplasts working again and to become photosynthetic again. So this is a sort of an ongoing process that has not led to a stable, to a complete stabilization of the chloroplast, or at least not yet. And here is another one, and in this case, it is it's an organism with the fucoxanthin, and its symbiont that's giving the color is actually a brown alga. It's actually a diatom that is captured and kept there. And here there are several cases like this one, Cryptopteridinium foliaceum, that has completely stabilized its chloroplast. You can have it in culture. We've had this in culture for years, and they don't need to be given any diatoms to eat, so the chloroplasts are completely stable. What is very unique in this situation is that you have the typical nucleus of the dinoflagellate with the very large chromosomes like the ones you saw before. But besides this one, you have another nucleus that looks very much like the general eukaryotic nuclei that we find in most eukaryotes. So it has these two nuclei together, and they divide in a coordinated way every time that the cell divides. So they keep there, both of them, which is rather remarkable. So, and then there are these sort of units that represent the chloroplast, or what is left of a symbiotic diatom. It also has a very peculiar eye spot, which is probably the remnant of the previous chloroplast, the previous peridinium chloroplast that the ancestor of these organisms probably had. This species, Cryptopteridinium foliaceum, is a marine species. You see it a lot especially in brackish water, but there are other species that are close relatives to it, of the genus Durinschia, which are not only common in brackish water, but you see them all the time also in fresh water. So this is the kind of thing I normally tell my students when we talk about these things, that although these all may seem rather wild and strange, these events, it is not necessary to turn on the TV and watch the National Geographic to find these things. Just collect some samples and look in the microscope. It is very possible or very probable that these guys will be there. So, and we just didn't understand this when I was a student. This knowledge didn't exist. Another one that is very, very commonly in our freshwater samples is this one. It used to be Gymnodinium. It used to be one of those blue Gymnodiniums. Now it is known under this name, Nusutodinium. Nusutodinium is a name that was coined by Japanese phycologists, and it's actually coined after a Japanese word. It is based on the term that the Japanese was used for stealing. So the idea is that it steals the chloroplasts. It has its kleptochloroplasts. And when you look carefully at this image, at this drawing, you see here very clearly a dinoflagellate nucleus, what is called typically a dinocaryon, with a very large chromosomes, very large nucleus, very conspicuous, unmistakable even in even in the light microscope. However, when you look at these units, which are obviously the chloroplasts, what you see is not only the chloroplast is here, but there is also a compartment that is lined by a membrane that is limiting some cytoplasm on the side of the chloroplast. So the chloroplast is accompanied by something. And in this something, you actually see a nucleus. And not only you see a complete nucleus, you see another small compartment inside this inner part close to the chloroplast, that also contains DNA. And this is what is called the nucleomorph of a cryptophyte. So this is the remnants of a cell of a crypto, perhaps a chromonas or something close to it, that the cell, the dinoflagellate, ate, and he manages to keep the whole system functioning and being photosynthetically active for some time until eventually it is lost. Either it is digested or it is expelled, but the cell will eventually lose its capacity, lose its color, lose its capacity to photosynthesize. But while it is here, if we count how many genomes this cell has, it's actually amazing, because it has the genome that is in its dinocaryon. It has another one that is in the nucleus of the symbiont. It has another one reduced to 4 chromosomes that is in the nucleomorph. And of course, the chloroplast has its own prokaryote type of genome, and the mitochondria that we see here in the cytoplasm of the dinoflagellate will also have its genome that's derived also from a bacterium. And you can see that inside this compartment next to the nucleus of the symbiont, you also have mitochondria, different from these, that will also have its genome. If you count, it's 6 different genomes inside this organism. That's something. So you can see it here. This is from freshwater. This is a low mag of a swimming cell. On the right-hand side, you see a high mag, and when you see the deep focus, you can see that the color is more or less confined to these areas. So the cryptos are, of course, a bit misshaped once they are inside the cell, but you can see that they are like units inside the cell. You can perhaps see it even better in this one. This is a brackish water one, but it is another species of Mesutodinium. And inside the cell, you can actually see the individual, what is left of the individual cryptomonads that have been ingested and that will function as chloroplasts for at least a while until the cell has to feed again and replace them. That's why it is called, they are called kleptochloroplasts. So this is summarized in this scheme. There are a number of them. I think this is a simple one that reflects what is generally accepted as the path that have been followed in this transfer of photosynthetic capacity, starting with free-living cyanobacterium down to the different types of chloroplasts that we see in the algae. And of course, we could put the land plants over there as well, but they are not that different from basic chloroplast type that we see in the green algae. So the dinoflagellates, of course, are here, getting the chloroplast from a red alga, but they, some of the members of the group did not stay like this and lost this type of chloroplast and replaced it, in this case, with the chloroplast coming from a green alga, and they became green. That's what you have in this Lepidodinium, for instance. Others have replaced the chloroplast with an heterocont. In this case, it is a diatom, and you have this group to which Cryptopyridinium durinskia belong here. Others have gained the chloroplast from aptophyte. That's what's happened, for instance, in Carinia. Carinia is a species very important because it causes red tides, it causes intoxications in the Gulf of Mexico every year. And you have a very special case in Dinophysis and so forth. So several differences, several different routes for chloroplasts known in the dinoflagellate group. So what about where they live? We have talked about that. You find the most diverse or the greatest diversity of dinoflagellates actually in marine and brackish waters. You can find them in salt pans, even when the salinity is very, very high. So they are able to survive and actually thrive sometimes in hypersaline water, but you find quite a diversity also in fresh water. You can find them in the plankton, in the water column, but you can find them in the sediment or attached to the sediment. There's a large community of organisms that includes a lot of diatoms, but also a lot of dinoflagellates that will live, for instance, in the muddy sediment on the bottom of lakes or estuaries, and that will actually perform movements up and down according to the time of the day, or more or less synchronized with the tide. And you can find them also sometimes as epiphytes, or very often swimming among the filaments of algae or aquatic plants. Both in fresh water and brackish water, you see all of these. And you find them all around the world, so they are very widespread, from Arctic waters to tropical waters. Nearly all kind of waters will have dinoflagellates. Just the one mention that must be made about dinoflagellates, and that is that some of the species will occur in mass occurrences, so much that they can deplete the oxygen of an area, especially when these so-called blooms decay, and a lot of the oxygen is consumed in this decay of a lot of organic matter, and that in itself can cause mortality of other organisms. But not only that, some species will produce toxic compounds, and they can accumulate on the food chain and produce drastic effects, like the one we see depicted here with this mortality of fish. The mortality can be also of other groups of organisms, and of course humans can suffer from it if they consume either fish or bivalves or other kinds of seafood that have been contaminated by these toxins. So this is the spectacular look of what is called a red tide, which in this case is really red. In other cases, the accumulations of dinoflagellates will produce different colors. There are other organisms that produce red tides, but the dinoflagellates have the sad record of toxic phytoplankton species over the world, and these are therefore some of the main causative agents of the so-called HAPs, the harmful algal blooms. Now, this is almost exclusively, as far as dinoflagellates are concerned, an event that occurs in seawater. If you go for freshwater, dinoflagellates are pretty harmless, with perhaps one exception, one species that has been claimed to produce a toxin that affects fish. But even this has been very, very few times that it has been reported. Not the species is not that uncommon, but the event of toxicity to fish has been recorded very, very few times in freshwater. So most of these problems concerning dinoflagellates are happening in the sea, around the coasts of many parts of the world. In freshwater, as you probably know, it is the cyanobacteria that have caused the greatest number of problems. Another mention about lifestyle or relationships that are that dinoflagellates get involved in must include the mention of their role as photosynthetic symbionts in the corals, in several types of organisms, but certainly the most important of this in quantitative terms and in how they are the basis of a very rich environment are the corals, which take in their inner tissues those coccoid cells of dinoflagellates, which were called at some stage zooxanthella, and are very active photosynthesizers And in this environment inside of the host, they actually exude a lot of the photosynthetate that they produce, and therefore they allow the coral to grow much faster, much better, and become much healthier than it would be without the dinoflagellates. So this symbiosis is actually perhaps one of the most important worldwide in terms of its ecological impact, and has received a lot of attention, especially because, as you probably know, the rise in temperature in many waters where coral reefs are very important has led to the disengage of this symbiosis, because the symbionts will tend to leave the cells of the coral, to leave the tissues of the coral, leaving the coral without the photosynthetic capacity, and that leads to mass mortalities in the coral reefs. And the whole system collapses if this happens. So there has been a huge attention to this relationship, and along with this attention, there has been a huge attention on the primary group of species that is involved in the symbiosis, and which has been called for many years Symbiodinium. Symbiodinium, it's not easy to work with, but the amount of work that has been put into studying these symbionts has been so great that whole phylogenies have been developed, and the characterization of different strains has been put forward, so that the genus Symbiodinium, if you look for the genus nowadays, you will find not one genus, but you'll find at the last count that I made, at least 10 new genera are now recognized to be involved in these symbioses. So that's what's happening when you study something very deeply. You find a lot of variation, you find a lot of different stuff, and if you want to learn about things, life becomes more difficult. Now, dinoflagellates are not only involved in symbiosis, but their interaction with other organisms, sometimes it's not so friendly. And there are quite a number of different dinoflagellates which engage on parasitic relationships with other organisms. Some of them will do that to unicellular organisms, others will do it with multicellular organisms, notably with fish, where they can be a real nuisance, for instance, for fish farms, like the genus Amylodinium, for instance, and other dinoflagellate parasites. What we see here on the right is an example of the development of a parasite which is called Tintinophagus, that is growing and parasitizing the cell of a tintinid. Tintinids are marine ciliates with this lorica around, so the cell infects and is starting to divide, where using the material from the host, the tintinid, actually the poor cell keeps swimming for quite a while until it is finally destroyed completely, as the number of offspring cells of the parasite grows and grows and grows. So this is one example. Most of these examples are from the marine environment. You see here another example, a dinoflagellate called Aprozoon. One of the characteristics of many of these parasites is that they will have the capacity to produce quite a large number of cells, usually small cells that will be mobile and that will reach out to infect new hosts, and therefore thereby propagating the parasite. This one is a parasite of a polychaete called Nicomache, and you can see it's pretty nasty. You can see how it uses its weapon to penetrate the tissues of the host. Here's a little video that was published when this species was described, and you can see this nasty stuff over there. And yeah, Yeah. Okay. But in freshwater, very few parasitic dinoflagellates have been described, but there have been a few. Here's a case of two parasites of fish that have been described from freshwater. One of them was named Piscinodinium, has this type of life cycle with this stage that will attach to the tissues of the fish and absorb nutrients from there. This is called Idodinium, has quite a different shape, but it also will attack some fish and have its part of its life cycle attached to it. So, when we think of cell structure, we've talked about amphiesma. Here is an image to show the different looks that we get when the amphiesma is composed or has these vesicles that are underlying the plasma lemma, apparently or mostly empty, like we see here in this transmission electron micrograph. And the result is this naked-looking cell, or when it is when provided of a relatively thick plate made of cellulose or a polysaccharide close to cellulose, and then you can have something that looks like this when viewed in the scanning microscope. So this would be an armored dinoflagellate. This would be a naked or athecate dinoflagellate. This is one of the traditional differences when you want to recognize species. Another feature that is characteristic, we have touched on that, is the nucleus. Here is the large nucleus. Here we have a longitudinal section through a whole cell. The large nucleus with a lot of chromosomes, large chromosomes, is here. We can see the cingulum right at the middle of the cell here. We can actually see the flagellum, the transverse flagellum, lying in the cingulum right there. A lot of chloroplast lobes. Here we have the nucleus of another cell, a cell that actually had symbiotic, at least it looks like they will be symbiotic because they look rather healthy. These bacteria that you can see here, and it's not very clear with this magnification, but I can tell you that this row of bacteria is actually inside the nucleus, which is something rather unusual, but was found in this organism. So another aspect that is quite unique about the nucleus, not only the way it looks, but the fact that the histones, the kind of basic proteins that you see in almost all eukaryotes as part of the basic structure of the chromatin, the way that DNA will wind around groups of these molecules to produce the basic unit that then will lead to a further coiling of the chromatin, is not present in dinoflagellates. They certainly have association with other kinds of proteins, but not the histones, which is very surprising. And I'm still waiting to find or to read about a convincing explanation of how and why this happened and why they changed from the path that almost all other eukaryotes have followed. That's just another aspect in which dinoflagellates are surprising. Another aspect, or a couple of aspects that are also unique to dinoflagellates, is they have their own type of mitochondria. Morphologically, in the electron microscope, you can recognize them from having these tubular cristas. Tubular cristae are also found, for instance, in the heterocons, but these have a constricted base, which is very characteristic. You can see that in these areas here. So they get a narrow base at the place of attachment to the inner membrane. You can also see here these two dark staining and squarish rods surrounded by a couple of membranes, and these are longitudinally, they look like this. This is one of these bodies ready to be ejected. So it's attached to the surface, and this is what it looks like if you see it in a number of different angles and sections. So it will eventually attach to the surface and has this more fibrous part, and then has the main body made up of these parallel proteinaceous rods, and eventually it gets expelled from the cell, perhaps as a device either to provoke a jerk in the cell and get it out of harm's way, or in some cases it has been shown that it can actually harm another organism and perhaps even help to capture a potential prey. So these are the so-called ejectisomes, and the term that is used for the ejectisomes of dinoflagellates is actually trichocysts. Trichocyst is the same term that is used for ciliates, and perhaps not surprisingly, now that we know that dinoflagellates and ciliates are actually closely related phylogenetically, the general structure of the trichocyst that you see in a ciliate is pretty much similar to the general structure that you see in the trichocysts of dinoflagellates. So when we look at the general cell structure, we have here the typical dinoflagellate with the transverse groove and the longitudinal groove, with the transverse flagellum where you can see the helical path of the axoneme separated from a more straight path of the inner fibrous rod. So you have this membrane-like type of flagellum, and here we have the other more normal-looking longitudinal flagellum. But there are dinoflagellates that do not have these two grooves, and yet here you see a protocentrum, the apical part. The two flagella are coming out of the apical part. There is no groove, neither a sulcus nor a cingulum, and yet what you see here is exactly the same configuration as you see here. So it's like a singular flagellum without a cingulum. And for many years, you had a lot of, you could read a lot about a lot of speculation about which is the primitive form. And there was a group of researchers that actually did a lot of work on protocentrum, and they usually wrote in their papers that it was the most primitive dinoflagellate. I have to say, I always found very peculiar why, if this was the primitive condition, why would they have developed this kind of flagellum that looks very much like the flagellum that you can wrap around in the cingulum. And now you will see that it is not thought anymore that this is a primitive group. It's actually a derived group that lost the two groups that make up this constitution of the cell. So you can, you probably have seen this, but just to show you the way the transverse flagellum actually works or beats, you can see it right there. Relatively slowly in this amphidinium. You can also see longitudinal flagellum waving here. And in this protocentrum, you can see it there. So you see that the transverse flagellum actually turns around as if it was around the cingulum, even though it is on the top of the cell, on the extremity of the cell. So the longitudinal flagellum is just here. The transverse flagellum, you can see it undulating there. So the cell, the flagella have their own peculiar construction, not only in what we can see outside, but also in the way they are inserted in the cell. You can have a hint of that here. You see the flagellum going into the cytoplasm there, but there is an area, sort of a canal, from the place where the flagellum goes out of the cytoplasm until it emerges outside of the cell. This can be a bit complicated. Let's see with drawing. So this is the longitudinal flagellum. This is the transverse flagellum that is coming out in this way. The transverse does not have anything special drawn here. It has it, but it would be very complicated and would hide this area if we had drawn it. But in this case, we have drawn this membrane-bound compartment from within which the flagellum comes out. So it comes out of the cytoplasm right there, but then it travels through this so-called flagellar canal before it reaches the region where the flagellar canal ends, and there's a fibrous ring on this part, and then it gets out of the cell. So this existence of the flagellar canals is one of the typical features of how the flagella get out of the cell in dinoflagellates. You will remember that in many other flagellate cells, the flagellum just emerges right at the surface, like in the green algae, for example. So if we look at it in a bit more detail, here's the amphiesma, here's the striated color that surrounds the opening, and the plasma membrane goes over the amphiesmal vesicles right until it gets to this fibrous area. Then it continues without being underlain by amphiesmal vesicles. It continues inside, and then it is continuous with the plasma membrane around the flagellum itself. So this continuity actually is what typically defines this as the plasma membrane. And you can see that there are some invaginations drawn here from the plasma membrane, and that they are wrapped in a vesicle or in a group of vesicles. And this is what is called the pusule. The pusule is another organelle that is typical of dinoflagellates, and it can come in many forms. We will not go into details there, they can be quite complicated, but this is certainly one of the forms. What is characteristic of the pusule is that in the end it's a diversion of the flagellar canal, whatever shape it has, and it is wrapped in the vesicle so that you have two membranes close together, and some distance away you have a third membrane. So any particle that is traveling here will have to, from the cytoplasm, will have to cross eventually three membranes to get into the lumen of this pusular vesicle. And since it's open to the flagellar canal, it will be on the way to connect directly with the outside of the cell. What are pusules for? Well, the most convincing, in my mind at least, function of the pusule seems to be osmoregulation, especially because unlike what you see in most freshwater organisms that have a flagellum, so that have a part of the plasma membrane that's exposed to the outside, in those organisms, if they are in freshwater, they have a problem because the osmolarity of freshwater is smaller than the osmolarity inside the cytoplasm, which means that they are in a hypotonic medium and the cell will tend to swell. Why doesn't it swell? Because in most of these organisms you have contractile vacuoles that are pumping the water out continuously. Dinoflagellates don't have real contractile vacuoles, so they have to solve the problem in a different way, and probably the pusule is the most significant system they have for coping with this difference in osmolarity. Although there still needs some research to get all of the functions of the pusule set once and for all. So it is still a bit of an enigma, everything that it can do. Very quickly, without wishing to become too complicated, we can see here the two flagella seen from the left side of a cell. We can see the bases of the transverse flagellum, the longitudinal flagellum right here, and we can see that there are a number of roots, microtubular roots attached to them. In this case, there are in fact 4 roots. Let's see if we can see them. So if we simplify the drawing a bit, what we see is that there are 2 microtubules coming from the association with the transverse flagellum. One of them runs alongside a fibrous root, striated fiber. The other goes on its own, but then it nucleates a row of microtubules that we can see there, and then associated with longitudinal flagellum, you have typically this multi-stranded microtubular root that will go down on the cell, usually underneath the sulcus, and a single microtubule that appears only in some groups of dinoflagellates, like typically the thecate ones, and that appears on the opposite side from this multi-stranded root. So this is a general view, and then there are variations which may be significant at a more lower level of division inside the dinoflagellate group. Another interesting thing is how they divide, particularly how the nucleus divides. The nucleus, in the case of dinoflagellates, does not lose its outer membranes, so the division is, from that point of view, a closed division. Closed mitosis is the term that is normally used, although I have some difficulty in using the term mitosis because we don't see the clear phases of mitosis that we see in most eukaryotic organisms. But in any case, the division operates through the development of these tunnels, cytoplasmic tunnels that will cross the nucleus. They are themselves lined by the nuclear membrane. And what we can see here in this spectacular micrograph is it was taken quite a long time ago by these two persons, is that the part of the chromosomes is attached through these membranes, through the nuclear envelope, with a row of microtubules that cross the nucleus inside this cytoplasmic channel. And these microtubules will be drawing the chromosomes to one side or the other side along this path, even with the presence of a nuclear membrane around. So it's a closed mitosis, and the microtubules in these channels will direct the chromosomes to one side or the other. So they have an old-fashioned 3D reconstruction here made out of some kind of plastic or plaster that was built layer after layer from section after section of observations. What does it look like when we see the cell division? Well, there are basically two types of cell division in dinoflagellates. One that is, you can say it's semi-conservative in the sense that the amphiesma, which in the case of this organism has visible plates. You can see plates very clearly up here. You can see the cingulum there. You can see the sulcus there. There's actually an active sulcal flagellum there. You can also see that there's an active singular flagellum right here. And the cell is dividing. So what we see is that this part of the plates of the amphiesma stays with the cell that is forming on the lower side, whereas the upper part stays with the cell that is forming on the upper side. So the cells will share the theca. Each one of them will have half and will have to produce the other half. This is one of the ways, a little bit more advanced here, the same division close to the end, close to the complete separation of the cells. You can see very clearly the part that has the theca inherited from the mother cell and the part which does not have theca yet and will have to produce it. These parts here are still naked or have still very little material inside the amphiesmal vesicles. So this is one of the ways, one of the themes for division. The other common theme is to have the cells stopping before division, losing the flagella, and then dividing in an immotile stage. Here we have a division, what's called a division cyst, with two cells already formed inside, waiting to mature and for the outer cover to become soft enough so that they can leave. There can be two cells in some cases formed like this, but there can be also 4 cells or even 8 cells. In this case, you have 4 cells that are ready to come out of the mother cell, a mother cell wall on the left, you have 8 that are preparing to leave this division cyst on the right-hand side. So in both cases, it's the same species, which was named actually recently, Torvelia renoi, and you can see how the cells become more and more active until they will rupture what is left of the outer part, and each one of them will move away in a different direction. So this is a different type of cell division in which the amphiesma, the whole outer part of the mother cell, is not going to be used, will be just left behind. Okay, we are not going to wait for all of them to disperse. I think we've seen enough to understand what is going to happen. So we move on. So also sexual reproduction you find in Dinoflagelates. If you go for books, say more than 30 years old, you will probably read that sexual reproduction in Dinoflagelates had not been demonstrated. But that's not the case. In fact, there had been demonstrations which had been questioned before, but they were quite right. And now quite a number of species have been demonstrated to undergo a life cycle with cell fusion and with reduction at some stage, so complete life cycle with sexual reproduction. What we have here is what we could observe in one of the situations where we could follow the cells in the lab. If we joined 2 compatible strains, we would see that from the vegetative cells, some cells would become smaller and smaller. This happens because there's a number of divisions, and this division we saw before directly into 8 cells is a very efficient way to produce small cells directly from a single division of a relatively large cell. And these small cells, if they come from strains that are compatible with one another, may become gametes. And the gametes will be attracted to each other and will actually swim around each other in what looks like a kind of a dance. And if things go right, I don't know what they say to each other, but if the things go right, they will start attaching and eventually fuse and merge into one another, producing a zygote. One interesting feature about the zygotes is that they tend to lose one of the transverse flagellum of one of the gametes. There are a few exceptions to that, but they will usually retain the 2 longitudinal flagella. So if you see one cell with 2 longitudinal flagella, it is very likely that it is a zygote, a planozygote, because it has retained the flagella. And then from this zygote, several things may happen. One of them is that it undergoes reductive division and produces vegetative cells. Another is that it may form a cyst, develop a wall, and then depending on the species, it may look very different. In this case of this species, it formed these projections that were more or less ramified, and it becomes a resting cyst. This resting cyst that clearly comes from a zygote is often called a hypnozygote, a resting zygote. And then from here, you can have reductive meiosis directly into a vegetative cell, or you can have the release of a cell, which sometimes actually comes out with 2 longitudinal flagella, hinting that there was no reductive division yet, and then have the meiosis, or whatever you want to call this, and become vegetative again. So there are a number of possibilities in these life cycles. You can see a little bit of the fusion process. Here are potential gametes swimming around another, and here's the beginning of cell fusion. We won't go into details here, but you can see that the cells are united by a little globule that is projecting from their ventral side, and that may be quite significant in the establishment of the compatibility between the strains. Here is division in the stages that are more advanced. The one on the right does not want to move. Apparently the computer is getting tired, and perhaps so are you. So I will stop this one in a moment. I think we are having a problem. Why can't I move this? Oh, it's not responding. Just let me try to get it back again. Okay. So the cysts are often made up, the wall of the cysts is made of a very resistant material, and this very resistant material is often capable of resisting the geological times, so it becomes fossil. And it is probably worth mentioning that these fossils are very much used by geologists because they've learned to relate the conditions in which the fossils were formed with the conditions of the environment. And this has or has had in the past a very significant practical interest, which is it would tell people looking for oil in marine sediments, it would tell them where it would be worth spending quite a lot of money trying to dig in and looking for the presence of a significant oil deposit in the ground. So they were used for this for many years, and so a lot of knowledge actually accumulated about these cysts. So there are people that can say all kinds of things from a group of cysts of this, like this. Each one of them has a species name, and so they have all kinds of inferences from this. And I will end this part of the talk with this image showing what I think is a rather beautiful display of bioluminescence by one dinoflagellate, in this case a marine dinoflagellate. Bioluminescence is found in several marine dinoflagellates. It's not a common thing in freshwater at all, but it is certainly something that must be nice to look at. So I will make a break here, and I would like to hear about what you think or what your prospect is.
Until now, we have no questions.
We have a question?
No. Until now, none. We can make a break of how many minutes?
Well, let's see. Why don't we stop for 15 minutes? Is that okay?
Okay.
So it is now almost past. So I would say when it's a quarter before the hour, we'll meet again.
Ok, perfect. Pessoal, então nós voltamos dentro de aproximadamente 15 minutos, lá por volta das 10h45, ok? Aproveitem para alongar um pouquinho, tomar um café, né, comer algo e preparar para a segunda rodada, para a segunda parte da nossa aula aqui sobre os dinoflagelados. Até mais então, pessoal. Olá, pessoal, estamos de volta. Deixa eu chamar o professor aqui novamente. Olá, professor Antônio. Novamente estamos aqui, espero que todos tenham descansado. A professora Vera fez um questionamento. Não sei se você quer colocar agora, verificar agora, ou colocar depois.
Podemos dizer alguma coisa sobre isso? Shall I switch to English so that I'm sure that everybody can understand? So what is being asked is about the puzzle. How spread is the puzzle in freshwater dinoflagellates? I would say that all dinoflagellates that have been examined ultrastructurally have shown to have a puzzle. The extent of the puzzle is very variable. In some cases, it actually fills quite a lot of cell space. In other cases, it can be rather discrete. So I would say that most things about the puzzle are rather enigmatic. One of the reasons we think that it will have to do with osmoregulation is that this arrangement of several membranes and a vesicle in between is perhaps appropriate for this pumping of water without being involved a contraction, as in contractile vacuoles. But even in the normal contractile vacuoles, there may be a contraction, but there is a pumping of water, an accumulation of water inside the vesicle before it contracts. So some of the similar mechanism may be involved here, and then there's an opening that is always there through the flagellar canals to the outside. Of course, this is probably not the only thing that puzzles can do, because the marine dinoflagellates also have puzzles. Sometimes even some significantly large puzzles. So in that case, it's difficult to argue that they were doing something concerned with osmoregulation, because the marine environment is not challenging in that respect, since the salinity is either similar or sometimes even higher than the salinity inside the cytoplasm. So this osmotic problem is by no means the same in the marine environment. So in those cases, perhaps the selective excretion of some products from the cell may be one of the functions. Some people have suggested that they might be actually involved in ingestion, but I don't think that if it happens, it will certainly not be a general thing. I don't think it is that is the actual function of the puzzles. I would think of them much more as devices for expelling something out of the cell than from uptaking into the cell. I don't know if this responds to the question.
Vamos aguardar. Let's wait, and then maybe Vera could complement the questions or clarify us if you could answer the complete answer for the question. Well, let's continue.
Okay. So I've thought about what might be interesting to some of you, and I thought that maybe a little bit more about this nutrition modes in dinoflagellates could be of interest to some of you. Some of you may have seen some of this, some of these videos before. Others perhaps not. well, I hope it can be of some interest to see that also in this respect, dinoflagellates are unusually diverse, more diverse than we are used to thinking about when we think about other groups of algae. So we've seen that there is autotrophy and heterotrophy in dinoflagellates. Some of them have chloroplasts, others do not have chloroplasts at all. Some do not have chloroplasts, permanent chloroplasts, but they can get them and use them for photosynthesis for a while and then have to replace them. So there are quite a lot of situations in dinoflagellates. So we range from the photosynthetic species with permanent chloroplasts that have are competent in terms of autotrophy. I have put more or less there because sometimes they will need something and then they will be auxotrophs to something. But it is true that you can grow in synthetic media dinoflagellates, some species, for quite a long time, and they seem to not need anything else. It is also true that sometimes you grow a species for quite a long time, and it seems to be okay, and then all of a sudden you start having problems with the cultures, and the culture, sometimes you lose it, and it is not easy to understand why. One reason might be that the cells have still some component that they pass on from generation to generation until that component becomes so scarce that the cells would need to get it from an outside source. And if they don't have it because we have not identified what it might be, then they will eventually die. So all kinds of these situations may happen, and we don't always have a good explanation for them. So other species are completely heterotrophic. They never retain a functional chloroplast. They just digest whatever they take in. And quite a number of species actually combine photosynthesis with food uptake, and they are called the mixotrophic species. Mixotrophic may mean several things. It could mean that you combine photosynthesis with the simple absorption of organic molecules from the environment, but it is sometimes used, or very often used, to signify the combination of food uptake, or at least utilization of organic food together with the capability to photosynthesize. There are different levels in these capacities. I will talk a little bit about that in a moment. Let's just see what kinds of mechanisms do dinoflagellates use to feed on particles. You will see why I'm not using the term uptake particles, because uptake is not always involved, although there is definitely a particle, or usually a prey organism, involved. So we have, in general, 3 different modes. The direct engulfment, which is probably the one that people will think about first, which is the cell ingesting, involving, uptaking a complete prey or a complete organic particle. There's a mode of feeding which has been called pallium feeding, in which there is really no ingestion, in the sense that the particle is taken inside the cell, so there really is no uptake of the particle. We will see a little bit more about that. And then there is the so-called peduncle and tube feeding, which is sort of a vacuum cleaning system that can uptake sometimes the whole cell, but very often only the inside of a prey, leaving the outside. So it's like sucking through a straw, or what kind of comparison you can make. So we'll just see a little bit about these three processes with a couple of variations. So starting with the direct engulfment, this is of course what it looks like in a number of examples from Dinoflagelates. Here we see a cell uptaking another cell through the apparently through the hypocone, perhaps through the upper part of the hypocone. Here we can see the evidence of uptake, where we can clearly recognize a whole cell. In this case, it was a dinoflagellate, clearly because of the cingulum here, that was uptaken by another dinoflagellate. Here it's other types of unmistakable objects. These are diatoms, diatom fossils, which normally can stay quite recognizable inside a cell even after being uptaken. So these are evidence of direct engulfment, food uptake, and even of quite large items by some dinoflagellates. One case of direct engulfment, which I think is rather interesting, is the case where we have this species, Fragilidium, which is drawn here. It's actually a thecate dinoflagellate, where you see the plates of the amphiasma quite clearly. You can actually order them in a way that is useful for the recognition of the species, because they are very regular and relatively few and relatively large. So this is a situation where we would not directly expect, without some experience, that this might be able to uptake something. Another reason why it would be surprising to expect this cell to involve in food uptake is that it is actually, it has chloroplasts and it is actually photosynthetic. The case is rather surprising because you can actually grow it in culture without giving it any food at all. So it is fully competent in terms of photosynthesis. It can live as a photosynthetic organism alone. What is also surprising is that you can stop giving it light, give it food, and it will grow as well. So it can also function as a complete heterotroph if it doesn't have light to perform photosynthesis. So it can do both to what you would say the extreme level, can be from the extreme photosynthetic organism, extreme autotroph, to the extreme heterotroph. So it is quite a remarkable organism. What is also remarkable about this is what it's eating. So the Fragilidium is the small cell that you see here, and it is now attached to another dinoflagellate, which is this large one, that is a marine species of Serratium. And here you can see how the cell is modifying its shape, and attached to one of the horns of the Serratium, it is what you see moving now is the actual speed. The speed has not been modified, and it is visible how the cell is bending that horn, bringing it closer to the cell body of the Serratium. And now, a little bit later, 12 minutes later from that, we see how it is already spreading around the main body of the Serratium. Now it's almost closed it. There's a little horn. One of the antapical horns is still sticking out, which you would think is a problem, but it is not a problem that this cell cannot solve. It just takes some time. So I think it is starting to be visible that it will bend the whole structure to bring the two horns closer together. And I remind you that this is a cell. There's nothing attached. It's just one cell. performing this. You can see on the left side of the bighorn that the flagellum is still there and it's still moving. To have an idea of what the cell is doing, you can think about the volumes. The serratium, the cell that is being ingested, is about 5 times the volume of the cell of the flagellum, the one that is ingesting it. So it takes some time, it takes a little bit of effort, but the whole serratium will end up inside and eventually be digested by this relatively small cell of the flagellum. We don't need to wait for it at normal speed. I will just speed it up a little bit here. You can see it takes a while, but eventually the serratium is completely involved, completely ingested. And at this stage, what you see here is this is probably the flagellum, but you see the plates also, the cell cover is being shed by the flagellum. You can see it very clearly there. So it's sort of the coat, the outer part of the cell has become so stretched that it actually needs a new one. So it sheds the coat, and it will now quietly digest what it has ingested and produce a new amphiesma. So this is some images of what is going on. In this case, it was a serratium that was being attacked at the same time by 2 flagellums. And this has been stained with calcofluor. Calcofluor will stain the cellulosic plates so that you can actually see at the same time the cells are stretching to start ingesting the prey cell. The plates actually become separated, so it stretches, and the old amphiesma gets swollen and the plates spread apart. So that's this very, very special case of this flagellum. Flagellum is a marine thing, so it is a good example, but not an example from freshwater. But here we have an example from freshwater mixotroph. This is this unicell, and you can see these colored organelles here. They're actually chloroplasts. They look like chloroplasts in the light microscope, and when we section these cells and look at them in the electron microscope, they look just like typical peridinian chloroplasts with a perfect arrangement of thylakoids, so they look completely functional. Whether or not they are really effective is a question. Maybe they are not so, they look functional, but maybe they are not enough for the cell, because the cells are very active, very voracious feeders. And they feed on several things. Here we had cells of chlamydomonas, which was being the prey, but we could also have cells of Scinadesmus. So it seems it is not very specific about its prey. We will not see the process moving, but we have a still image here of a round prey cell, some sort of greenish cell that was about to be taken up. And we can see that the cell has this projection at the moment of feeding, and this more visible part of the projection is actually dragging a layer of cytoplasm whose limit is here. And so this flap will turn around the prey cell and drag it into the predator cell. So this is the way that this, let's call it Bernardinium, eats. So it is a total, it's a complete engulfment, although in this area, the mechanism for the stretching of this part actually involves a similar machinery, cell machinery, than the process that we will see last on this presentation, which is the tube feeding. And you can see here, there's a row of microtubules. There's another one here, but this is related to the flagellar apparatus. But this one is not. It is leading exactly to the place where a little tube or a little column of cytoplasm will come out. You can see it here in higher magnification. Here are the microtubules. Here's the fibrous material that's coming down from the area where this cytoplasm will attach to the prey and then start taking in the contents of the prey. You can see also these vesicles that appear in the electron microscope with this dark appearance, and they are normally present in these systems when they are actually functional and may be involved in feeding. So it is very likely that all of these are quite full of digestive enzymes that will participate in the process of digesting prey at some stage. By the way, the puzzle, you can see it here. In this case, it's a tube. You can see part of it here, but in other areas of the cell, it will fill up quite a large portion of the cell as a convoluted tube that connects to one of the flagellar canals. You can see the flagellar canal right there, in the transverse flagellum in that case is visible in this place. So that's a mixotrophy, and this is just to show you that the part that extends is actually supported by this long row of microtubules that will eventually extend right here outside of the cell. So a row of microtubules, that is something common in many dinoflagellates. It seems here involved in the food uptake, but not in the usual way that these things perform in other dinoflagellates. So here we have a different thing, a drawing showing what is called pallium feeding. And what is pallium feeding? What you have here is the cell is exuding a very thin layer of cytoplasm, and this very thin layer of cytoplasm has spread until it has enveloped completely a cell, a hypothetical cell of a prey. In this case, it's perhaps a diatom that is being enveloped. There could be something else. So what is shown here is the way that these dinoflagellate cells behave when they are faced with a potential prey. And in this case, the potential prey is a chain, is a colony of the diatom Chaetoceros. Chaetoceros is notable for having the long spines attached to each one of the valves. So with these long spines, they are not really the kind of thing that looks easy to use as food. It's not easy to ingest. It becomes very large, very wide. So most predators will not be able to do anything with it. But this dinoflagellate is here seen swimming around this chain of the diatom as if it was sizing it, as if it was deciding how to handle it. So it goes from one end to the next and goes around in several ways and goes around and around and around until at some stage it will sort of shoot something into the chain and attach to it, connect with it. And then it starts dragging it around, so it is already connected. And what happens next is that it starts sending out this very thin layer of cytoplasm that starts spreading and spreading and spreading around the prey until it envelops completely, or at least a part of the chain, and starts digesting it in place. So digestion happens without ingestion of any cell. It just envelops the whole thing and starts digesting it in place it is possible to see how the chloroplasts of the prey, of the diatom, start losing their appearance, losing their color, losing their shape. And then when it's done, when the digestion is done, it will bring back its thin veil of cytoplasm, and that's how it fed. You can have an idea of what the cytoplasm looks like in this video. The cell has attached to the bottom of a counting chamber. The reason why it has attached, it's probably because it has been fooled by a layer of organic material on top of the cover slip that is on the bottom of the counting chamber. And you can see it's turning around. You can see it has this sort of a filament that attaches it to the bottom, and you can see this column of cytoplasm that is moving down the filament towards the surface of this chamber. This is, again, real speed. It has not been modified in any way. It has now reached the bottom, and it will start spreading over the bottom, as it would start spreading over a potential prey if that was really what was there. So the cell behaves as if it is enveloping a prey and try to digest it. So that's exactly what it would do if it was actually an organism that it could consume. This is what it looks like at the end of this spreading period. So all of that is the pallium. You can see the edge of the pallium. There's a scale there, so you can have an idea of the size of it. In this case, it's a very probably unusual shape for a pallium because it became round. If it was a chain of diatoms, it will have enveloped the whole chain. And now some signal, or probably because nothing happens and there's not really anything to digest there, it starts pulling back the pallium. So that's what we're seeing now. Again, this is the normal speed. I will try to speed it up a bit. Yeah. Now I am moving the video forward, but let it move by itself now. So that whole portion of cytoplasm that had been taken out was now in the cell. And this is, of course, the real thing. We have a chain of these diatoms enveloped by this pallium, and it's being attacked by this protoperidinium. Protoperidinium is an heterotrophic group, and it was rather enigmatic for many years how these cells of protoperidinium, which are actually relatively large cells, you never see them with chloroplasts, so they are obviously heterotrophic. They need to eat. They are too big to be able to survive just by absorbing nutrients, organic molecules from the environment. There's not enough concentration on the environment for that. And yet you never see them with particles, organic particles inside. So how did they do it? This was completely unknown until the mid-1980s, when somebody saw for the first time this mechanism of using this thin layer of cytoplasm, the pallium, to feed. So that is pallium feeding in Protoperidinium. This is a marine species, but one may think, is this completely exclusively a mechanism used by marine species? Here we see the drawing of what we were looking at a moment ago. And here we see the basis, or the support, the cytoskeletal support for the pallium, which is this row, this row of microtubules that extends in the pallium, when it spreads towards the prey, at least part of the way. So a single row of microtubules will be used to help spread this cytoplasm out of the cell. And these are several images. Here the prey is another dinoflagellate, it's a serraceum. Here it is diatoms. So several species, either of Protoperidinium or of species of this group of called Diplopsilus, will use this system of pallium. This has actually, it stands as a mechanism that one can trace to a group, a phylogenetic group of organisms. If you look here at the areas that are within the rectangle, here you see Protoperidinium, and here you see Diplopsilus, and all of the species here belong to these genera that belong to the same group. So here again, Diplopsilus, other things that are now called with other generic names, but were at one stage called Diplopsilus, and they share this general shape of a lens, and Protoperidinium put together in a group. So it's a case where apparently a mechanism of feeding has evolved in an ancestor, has been inherited in a whole group of organisms. The group actually corresponds to what we now call family Protoperidiniaceae, and the highlight here is for this species, Colchicella acuta. You can find it in the books under different names, Diplopsilus acuta, again the name Diplopsilus, or Entia acuta. And you can see that it is grouping together with a group of Protoperidinium. So again, it is in the same group, it is in fresh water, and it feeds with the pallium. We know that because it has the structures, and we know that because we have actually seen it. So again, a freshwater species that uses this mechanism. There are not many, perhaps this is the only one that really does that. The species looks like this in freshwater, and you can see there's an opening inside the sulcus, and that's where the pallium comes out, and that's where probably where it will go in after feeding. And here is a transmission electron micrograph of the same organism. Here's the sulcus, and here is the pallium on its way out or in, I don't know, but on its passage through the sulcus and the way that it normally feeds. So that was pallium feeding. Another way of feeding, which is in some ways quite interesting, is what has been called peduncle feeding and tube feeding. Some people use it as meaning the same. They don't mean quite the same, because in one case you have what you could say a column of cytoplasm attaching to the prey. In other cases, what you have is an actual tube that is deployed by the dinoflagellate against the prey, or against the part of the prey that can be sucked in. So what can we say about this? Here, the example is actually from freshwater. This is a freshwater organism. It used to be called an Amphidinium. Now it is in a genus of its own. It's called Algas. And you have seen this image before. Here we have the part that is involved in the feeding. And you can perhaps see this line here corresponds to a microtubule. So there's a row of microtubules up in this upper part that helps this cytoplasmic column be extended. We have some fibrous material here around the exit point, and this is the part where the food will come into the cell when it starts feeding. We have tried to reconstruct this part, and the result, a 3D drawing trying to represent this whole part, shows exactly this like a cylinder of microtubules that then spreads inside the cell and continues for a while, and that's probably the sliding of these microtubules against one another will be what is driving forward and backward this tube-like thing, or this column-like projection of cytoplasm. You can also see the flagellar apparatus. I will not go into details there. Let's just look at the feeding. This is what it looks like when you photograph it in the microscope. This is the area that gets extended, and this is feeding on, in this case, it was a green alga, spermatozoopsis, that was being ingested. And this is the back part of it, the pointed back side of it, that is all that is sticking out. And here is a view of a cryptomonad being ingested. The cell of the dinoflagellate is being seen from the top, from here, and you can see how the cytoplasm of the prey is streaming in through the middle of the epicone of the dinoflagellate. So we can see it a little bit in this video. You can see the cell, transverse flagellum is very, very clear. You can see the cell is quite small. The two flagella are very clear, and you can see that there is some food inside there. And we'll see how it's feeding on a crypto. It has attached to the cryptomonad, looks actually like a cryptomonas. And what you can see there is that the cryptomonas is stopped moving. So there seems to be like a paralyzing effect, and the two flagella of the cryptomonas will stop completely when the attachment takes place. And now the cell is clearly busy doing something. The dinoflagellate is doing something with the prey. You can see that there is no uptake yet, and the cell is not really spreading around the prey. So what it is doing is it's drilling its way into the contents of the cryptomonad. Now you see there is a stream of colored material go into the dinoflagellate as it is sucking the contents. So it does not ingest the whole thing, it just drills a hole on the cover of the cryptomonad and starts sucking in the contents. This is possible to see, easy to see, because of course the cell, the dinoflagellate, is colorless when it doesn't have food, and the contents have this characteristic color, strong color of the dinoflagellate. Here you can see the flagella of the crypto. They are like paralyzed. The attachment was on the back of the cryptomonad cell, and the flagella of the crypto are just sticking there without any movement. The cell is obviously in the phase where it's doing something, trying to drill a hole through the cuticle of the cryptomonad, and until it does, there is no uptake. It didn't take long to take this one completely. This is the end of it. It usually leaves something behind, and this something behind must contain a little bit of cytoplasm and probably most of the cover of the cryptomonad. Here we have an apical view that shows where the food actually travels to the inside of the small prosoalx. It's an apical view because the cell was swimming, but it was swimming against the cover slip. So it was swimming in the same place, which made it easy to focus the microscope and follow what was going on. And you see the stream of contents of the crypto being driven inside the epicone of the dinoflagellate. It's swimming all the time. This one it was the occasion of seeing the first attachment, because another cell came and attached. And in the first stages of attachment, you can actually see the appendix the feeding column, the feeding tube, being still more or less spread out, or stretched out, not spread, stretched. You can see it there by stopping the video. You can see how it is attaching to the crypto. And now the situation, the two cells will start taking up the contents, the inside of the cryptomonad cell, and the end result 5 minutes later is this, and you'll see they will leave a little bit behind. Only the content was taken, in this case divided between the two predator cells. So that was Curso Algas. If you think that's awful, Curso Algas is child's play compared to these guys. These are the Fisteriaceans. Some of you may remember a big fuss some 20 years ago about Fisteria. When it was described, you had it in the news, you had books written about it, people were referring to it as the cell from hell and all kinds of things. Apparently, much of that was due to some unfortunate mistakes, especially concerning some of its toxicity, although some people still claim that it does have toxicity, but there was a lot of fuss about this. But one thing is certain, and that is the way they eat. You can see on the images below a cell of Fisteria feeding on a fish red blood cell, and you can see on the figure on the left the feeding tube stretching from the lower part of the Fisteria cell to the red blood cell, and it is being sucked in until it almost disappeared on this side. The feeding tube can be seen here in this scanning micrograph. So this is the flagellum, the longitudinal flagellum. Actually, there are two of them, which makes me think that this would be a planozygote, so sexual reproduction. The transverse flagellum, you can see it here undulating, and this stretched feeding tube is here. This is taken, I believe, on the tissue, the gill tissue of a fish. So this one was actually attacking the gill tissue of a fish. So this is what it looks like in the TEM. This is from Fisteria, this marine one, but this one is from freshwater, and it's actually one of the very common colorless dinoflagellates with theca that you can see in freshwater. And you can see the same structure of the feeding tube. The feeding tube of Fisteria is right there, and the feeding tube of this freshwater guy is here. And you can see it's actually a tube. If you look at it in high magnification, you can see a membrane going around on the outer side and turning into the inside and continues on the inside. So cytoplasm is here only in this hollow cylinder, and this is just the route to suck in food to the inside of the cell. And this whole area that I'm trying to delineate here is actually food that has been uptaken. So the nucleus is this very large object that's on the hypocone, but the rest here on the epicone is totally filled with food that it has uptaken from its prey. And its prey in this case is a nematode, hundreds of times its size. Well, of course, it had a little help to be able to handle the nematode, but in any case, this is a bit of an inversion of the relationship between prey and predator that you would expect from the sizes alone of the organisms involved. The organism was first called Tyrannodinium, well, it used to be called Peridiniopsis beroliniensis, then it became Tyrannodinium berolinensis, and then from comparison with an older name from an older described species, it is now called Tyrannodinium edax. So this name is the microscopic counterpart of another TX, which is the T-Rex, which was supposed to be a terrible predator in its own time, feeding on just about any other thing. Well, T-Rex, apparently, we only know it as fossil until somebody does something close to the films and starts reviving these things. It's a fossil. And but T-Edex is real enough, and it's all over. Here's the look of it from the TEM. This is in a Pfisteria, and this is in the Pterodonu edex. And you can see that instead of a single row of microtubules, it actually has a whole array of rows of microtubules. And it is undoubtedly the sliding of these rows of microtubules along one another, probably added by the sliding of the individual microtubules within a row, that gives a very, very dynamic and very strong capacity to this feeding tube to adapt and suck in whatever is available. And this is what our freshwater species looks like. In the light microscope, you can see a huge nucleus with large chromosomes. This was probably a dividing cell. You can see that some of the chromosomes are actually aligned. You can see the remnants of a food vacuole, probably a digested one waiting to be thrown out of the cell. And here you see what it looks like if you manage to prepare it adequately for SEM. So it has actually a theca, has plates, and that made us, wonder how it could feed. Well, it can feed in this way. So this is the drawing that shows the process of feeding. This is a nematode. The nematode had a little hole on its surface, and since the contents of a nematode seem to be in a pressure, part of the gut of the nematode is actually coming out here. And the cell is attracted to what is being released here. So it has a very sensitive chemosensory system that draws it exactly to the point where something that it might find useful is coming out. And during this process, it swims in a very characteristic way, swims around an axis that goes through the middle of the cell. And then at some point, it will shoot into the cell, perhaps by some ejectosome or some group of ejectosomes. We couldn't make sure of that, but it shoots this cable that will attach it to the food. And afterwards, it deploys the feeding tube. And it starts feeding, and in the end, it will swim away. And sometimes at this point, it actually is arrested for a while by the original cable that you can see here when it's trying to swim away. So this is what it does, and I'll show you a little bit of the movie that we made at the time, and we called it this. I think this film, which some of you might recognize, had been on for not very long, so we decided to call it like that. So this is the cell, colorless, Peridiniopsis is what we called it at the time, and swimming peacefully, looks rather harmless. You can see no chloroplasts, completely colorless. It's actually quite small. You see the scale of 20 microns, so this particular cell is not much more than 20 microns long. And now we see some action. That is a rotifer that was damaged, and you can see a cell on the left. You can actually see some food uptake there as a feeding tube. But here what we were trying to show is the characteristic swimming mode. You can see the bacteria here. Bacteria are the first to arrive. They have a very, very sensitive chemosensory mechanism. So as soon as something is leaking out of an organism, they will be there. But shortly after, will these cells. Here you can see how accurate this mechanism is. This nematode was injured with a pincet, which means that these two cells are attached on one of the sides where it was injured, and this cell attached exactly on the other side. But you can see it's minimal, the damage, and there's nothing leaking out. So you can see the cell has deployed its feeding tube, and it's actually pulling. It's pulling, trying to get something out, and nothing is coming out. But still, it is that point that was injured. It was not here, it was not there, it was exactly there. You can see the movement of the particles around. This is because the transverse flagellum keeps moving all the time. And that was the reason for using a nematode much larger than the cell to try and film these things. Because otherwise they're swimming all the time and you cannot follow them. You have to give them something that is much bigger than themselves, so that they will stay in the place where we want to film them. And it's trying, and it's trying, and it's trying, and it's stretching. And you can see the empty area up here. It's already open, waiting for something to go in. There's a continuum between all of this, you see there. And still nothing has come. But after a while, you can see it was the right spot. Finally, something started to go in. So this is the very, very sensitive mechanism. Here is another freshwater species, which we decided to check to see if it would behave in similar way, although it has chloroplasts. Well, we joined it with Tyrannodinium, and the result was this. It didn't care whether it was a fellow of the same group or if it was something else. Here you can see very clearly the food uptake, the contents being taken in because they are colored. You can see how the chloroplasts start to fill up the upper part of the cell, the epicone of the cell. And actually, the chemical attraction made another cell come in, and the other one is bigger and actually drove the whole thing away. This is high mag on the nematode. So you can see how the food is actually being uptaken. You can see the feeding tube very well, and the flow, the speed of the flow. Here you can see how flexible the tube is. It actually wraps around the end of that structure. The whole upper part is already filled with food that has been uptaken in this way, but still it's getting more, it's sucking out more. And you can see the chromosomes and the large nucleus here. The flagella are still moving. You can see moving the longitudinal flagella from time to time on this area, and you can see the transverse flagellum moving, and sometimes you can see the movement of particles around that is drawn by the transverse flagellum. That's the edge of the feeding tube, quite flexible. And when we section this on the TEM, you'll see microtubules, these rows of microtubules going all the way almost to the edge of the feeding tube. And undoubtedly, it's all that apparatus of microtubules that gives strength and flexibility to this structure. So it is an essential feature of this group of organisms to which this Tyrannodinium belongs, but also this Pfisteria and a few other genera close to Pfisteria belong. It's a very well defined group. If you think of this feeding mode as a defining feature, the feeding mode that involves not only the mechanism of the tube and the cell machinery to form the tube, but also this chemosensory mechanism of finding exactly the spots where you have a probability of finding food being in condition of being uptaken. So that's yeah, that's another view of it. I think I will drive this forward a bit so that we don't waste too much time. Here is something about the mechanism. You may form your own idea of what is driving that, but you can see that it's leaking down here. So it sort of sucks in and then it comes down again and then it has to suck it again. But you can figure out for yourselves or have your own theory about what drives this, how this can be done. The cell is very versatile, and you can see this one is trying to feed on this place. There's nothing coming out. So it just, yeah, it's a little bit coming out, but not enough. So it will try again in another place. It will readjust itself, you can see, keeps the contact on the edge of the feeding tube and just tries another place. This is how versatile they are. And here is actually the attachment of this harpoon or cable or what you might call it. To my knowledge, this has not been shown by anybody else in any other occasion. You probably didn't see it the first time. We'll look at it again. Slow motion. Bang, there it was. And if you didn't see it yet, we will look at it again, even slower motion. Cell is turning around. Pow. There, you see it. So a couple of frames and that's it. That means less than 1/10 of a second and it's shot. And there is this attachment filament. That's what's keeping this cell from swimming away. It has to work its way away from the food because it's being held by the original filament that it shot into the food to start with. You can see it very clearly here. There it goes. So this is the group to which Tyrannodinium belongs. Tyrannodinium, at that time with the name Peridiniopsis berlinensis, is here. This is the tabulation, the epitheca, the arrangement of plates, and these are the variations that have been described from several species. Here's the Pfisteria and several others. This is Amylodinium. It's actually quite an important species for fish farms because it's a parasite fish, and several other species with several other variants in the theca. And here is what phylogeny says about them. You can see that they form this group, which we actually like to call with the name of a family, the Pfisteriaceae. Of course, using the name of a family to this group with this type of branching will make this group paraphyletic. But even so, this is such a well-defined group that I think there is some advantage in having a name for them. And this is a group that has this tube feeding driven by these array of microtubules, which is what has been called a microtubular basket. Now, a microtubular basket was first described in 1969 from an organism which came to be known with the name Cryptocodinium conii. And you can see all the elements here for feeding. You can see the different rows of microtubules that are exactly with the same relationship to each other as we find in Pfisteria and as we find in Tyrannodinium. You see it here sectioned longitudinally. In addition to the rows of microtubules, you have these vesicles, including these vesicles with dark appearance in the TEM, which seems to be involved in transporting enzymes, digestive enzymes. So everything that you could think of for feeding with this type of feeding that we see in the Pfisteriaceans. However, the organism was found by Jawornicki in 1962, and he described it as being mainly coccoid, so non-motile cells, so much so that he put it in a group of coccoid dinoflagellates, and he saw no ingestion whatsoever, so no tendency to feed. That was rather surprising until in 97 a paper came out describing for the species the feeding tube and how it actually was attracted and fed on dying cells of a red alga. So that made more sense. And here is again the microtubular basket and several views of it. So everything that we were expecting to see in the fusteriation. So is it a fusteriation? It's not easy to say because it's a very derived organ, is very specialized, and it forms a long branch in the phylogenetic tree. So long branches, as you probably know, are usually not very reliable. They are liable to all kinds of artifacts, but at least in this situation, for the first time in this paper by Orr et al. In 2012, it came out of in a phylogenetic tree generated from 8 genes. It came together with the group of the fusteriaceans. There is Cryptoperineopsis, Pseudophysteria, Physteria, and that for the first time to me made sense. So although there's a very long branch here, I would tend to think that this is the right phylogenetic position for this, because it has all the elements that we can see in the behavior and in the cell machinery for feeding in this particular way. And here we see the fusteriaceans together with Tyrannodinium. It was still named Tyrannodinium berolinense, a name that would certainly not be possible if we were in the 1930s. We would probably go to a concentration camp if we said this, but nowadays we can do it. So, just to finish with a note, this we have established this feeding mode as characteristic of this group of species, which we like to call the fusteriaceans. However, the microtubular basket, that is the cell basis for this type of feeding, is not exclusively found in the fusteriaceans. We see photosynthetic species here, which nobody has seen feeding, and yet it has its microtubular basket, undisputably the same type of structure, and it is outside of the group of fusteriaceans. It's in the neighboring group of the Pterocosphoraceae, and so it means that the ancestor that developed this type of machinery may have then evolved along 2 different lines. One of them that's stuck to this feeding mode as the main means of existence, whereas others turned into a primarily photosynthetic mode of nutrition. So things are always more complicated than we would perhaps like them to be. And this is a little fusteriation. I think it is probably a Cryptoperineopsis saying goodbye to you, at least as far as this presentation is concerned. So, I have a very small presentation. We don't have much time, so I will leave it to you to decide whether you want to continue with that, or you prefer to some kinds of questions, or what you want to do.
Vamos lá. Deixa eu só parar a câmera aqui só um minutinho. We have another question from Vera.
From Vera. Now, the proportion of species and freshwater that can be phagotrophic. It is, as far as has been demonstrated, relatively small, I would say. There are these hardcore predators that will be eating, and they need to eat. There are those that need to replace the chloroplasts, so all the species of Nusutodinium for sure will have to eat. I've never seen any evidence, for instance, that those that have diatoms as symbionts, the ones we see in freshwater, I have not found any evidence that the diatom may not be permanent and therefore they may eat. However, this has been described for some in the marine environment, for some closely related species of Dudinskia. So it is not impossible that it may yet be found. But when we come to Peridinium, for instance, the bulk of the species, I would say, if they ever eat, it's an exceptional event. We've tried to look at that, and I would say that almost certainly most of these guys don't eat, or if they do, it will be very, very exceptional. So I would say the proportion is relatively small.
Okay. Another question, people? Você preferirá continuar, dar sequência ou fazer outro intervalo?
Se fizermos um intervalo, estamos quase no final. O intervalo iria até ao fim. São quase horas. Eu só tinha na última apresentação, tinha feito algo muito, muito resumido relativamente aos grupos em água doce, pelo menos tal como os dividimos na última flora. Mas é mesmo uma coisa muito curta e rápida, portanto, se isso não for o centro do vosso interesse, então, não sei, não vale a pena.
Não, eu acredito que nós podemos passar, sim. E aí, então, só que antes nós temos mais duas perguntas do Cauli.
Olha o Cauli! Yeah. There is only one way to go about that, I think. The Peridinium umbunatum or Parvodinium umbunatum is a nightmare because it does not have characteristics that we can recognize in modern taxa. When we look at the original Stein's drawings, the epicone is longer than anything we find again. Later attempts to define the taxon always fall short of the variation that we see in reality. So this is unsolvable. The only thing to do is to, I think, arbitrarily define something that could fit the original concept and very close to what we now call Peridinium inconspicum, try to find a strain that matches this, that could be acceptable as Peridinium umbunatum, defining it as Peridinium umbunatum, getting the DNA out of it, getting a molecular characterization of it, and have it as a reference point. Because even trying to go for the original places, there is no single original place for Peridinium umbunatum. There were at least two areas where it was found, and it is very much arbitrary to go to one of those areas, get a culture, and say, oh, this is from the type locality, and so and so. This is all very uncertain. To me, the only thing that can actually allow us to move forward is to pick up something that we can all accept as representing umbunatum, and to represent the concept of umbunatum without changing from what has been used in the last 50 years or so. It has to be something very close to a concept of inconspicum. So picking on one strain for umbunatum, perhaps a longer one, a bit larger cells, because that's one of the few differences that one may suppose could help define species in this group. So a larger strain for umbunatum and a smaller strain for inconspicum, which was originally described as quite small, and being completely conscious that this is would be arbitrary, but then we would have a reference point molecularly recognized, preferably with a morphological and ultrastructural characterization for each one of these two species, and we can move forward. What people are doing now is there is variation in this group. And this variation, of course, we all want it to be recognized as species. And there are already species being described in this group within this variation. But this is being done without fixing the reference points, which means that we may run into trouble, into confusion in the future. So I don't see any other way out of that. We have to agree on taking a strain as a reference point, characterize it molecularly, as long as it is something that can be acceptable and does not visibly change the use, the traditional use of these two names, umbunatum and conspicum, then that would be a benefit for everybody, I think.
Perfect. Okay, let's go to the last part.
Yes, I'll try to be brief because people are probably tired and they don't want to hear any more about these things for the rest of their lives. So, about the taxonomy of freshwater forms, and this is just a very, very, very brief outline. This is the outline that we did on the latest freshwater dinoflagellate flora, and it has all the question marks and the problems that you may expect, and you may expect that some of these things will change. It was divided into 12 orders, the groups in freshwater, and they are here. The first order is a bit inevitable if you're making a revision. It's the Desmosmastigales, which is a very enigmatic group, which nobody has refound since I think from the original description. It was described as having a typical dinoflagellate nucleus. Apart from that, it has two ribbon-like flagella, and nobody really knows what else. It has no groups on the surface, so what is it? If somebody finds it, that would be very interesting. Otherwise, it's a completely doubtful group. Dinoflagelados, again, the cells look like amoeba. The only connection is the nucleus and a swimming stage, apparently within only one flagellum. So also very enigmatic. It has to be there because we can't put it anywhere else. But if somebody finds it, that would be very interesting. The Amphidinium is usually not present in fresh water, but at least one species was described in fresh water. And the group with Amphidinium species usually appears rather separated from all the rest all the rest of the dinoflagellates, the core dinoflagellates in the phylogenetic trees. So it seemed to be too artificial to put it into the Gymnodiniales as was done in the past. So we had to segregate it to Amphidiniales. We'll see what the future will say about this. The Gymnodiniales, you can see here, it has the largest number of species. The justification for this in the flora is that we tried to reduce the number of species that were obviously misplaced in other groups. And the only practical way to do this is to have a garbage can. And the garbage can was Gymnodinium, because it was the genus to which already belonged most of the uncertain things. So everything that has not been studied recently, we don't know where to put it. We just knew that they were not correctly classified in other places. We send it back to Gymnodinium. That's why there are so many in this group. But it also contains Nusotodinium with a new classification, and we actually added a few more that had blue-green or blue chloroplasts, or had at least been described with blue chloroplasts, which we think is very likely are other examples of this kleptochloroplasts that we find in Nusotodinium. Gonialocales is a group with many species in marine water. In fresh water, it has only basically two genera. It's Serratium and Goniaulax, only one species, very rare, exists because it has been photographed and it has been examined by SEM, but it is not something that we step on very often. So there's a very limited number of species. But the Serratium, of course, is something that you all know, especially in Brazil, especially one of the species that has been spreading quite a lot. The Peridiniales are, perhaps we should talk about Peridiniales together with the Thoracospherales. We're talking about the well-known Peridinium, the ones that have plates around the cell, plates that can be used to establish tabulation, an arrangement, then can be used for identification. The problem is that the traditional ways of classifying will display as very close together things that happen to be in different positions in the phylogenetic tree, and so some of them actually went up together with some marine species that form calcareous cysts, for instance, like the Scripsiella, and they are closer relatives to some of them. The Stresiales is a group that was revived based on fossils to start with. It seemed to be very, very rare nowadays. It was actually first a surprise when something that was obviously a Stresiales was found among living material, but now it's actually a large group, and even a very important group like Symbiodinium and the genera derived from Symbiodinium that are involved in the symbiosis with corals and so forth are placed in this group of the Stresiales. Toveliales, a different group, typically with a very, very peculiar eye spot that is made of oil globules that are not part of a chloroplast. It has both chloroplast-bearing species and species that are completely heterotrophic, and the cell cover is typically something in between the true thecate species and the true naked species. Phytodiniales, that's the name that we gave to the coccoid species, and it is likely that once they get studied by modern methods, several of these coccoid species may end up to be inside other groups, perhaps at least some of them within the Stresiales, but they need to be refound and they need to be restudied. The Gleodiniales contains basically one genus, perhaps two genera, with the cells that live in a coccoid stage or a palmaloid stage, if you want, surrounded by mucilage. There are relatively few species described. They are not easy to study, but perhaps they are a group, they are well on their own. And it needs to have some more observations. And the Protocentrales, there's nothing mysterious about it. It's very rare, this group, in freshwater, but it's very well known in the marine, in brackish water environments. The only thing is that in freshwater, there are only two species that have ever been documented and described, and they were found in Australia and in one of the islands of New Zealand, and they were, as far as I know, they've not been found in any other place in freshwater. So basically some of the most common members of the dinoflagellates in freshwater will be members of the Gymnodiniales. Here we have Gymnodinium palustre, or at least, well, I choose to be calling it that at this moment. Other people would have another opinion. And a species of these this species with kleptochloroplasts that are now called Nusotodinium aeruginosum. It's a very, very common dinoflagellate. And if you see it in the samples, if you look carefully at the samples, you will certainly find these very small chromonas-like cryptomonads for sure, because that's what they need to eat to get those nice bluish chloroplasts. The group of the Goniaulacales is, as I said, represented very, very common genus Serratium, very, very rare Goniaulax in freshwater. Serratium, the common species, Serratium irundinella on the left, Cornutum, very easy to recognize on the right, and of course the Fucoides, which you have spreading in Brazil or in South America in general, in which we have had here for quite a lot of years. You can see its characteristic short plate number 1 that does not reach the top of the apical horn. You can see it delineated here, and together with this shape without the shoulders that the Irundinella shows. Then you have this group Peridinialis and Pterocosphoralis, where you have these cells with the typical dinoflagellate morphology, and with the plates that are relatively few, relatively large, very constant, and so they lend themselves to classification. The plates are arranged in a, they are numbered and individually recognized, so you can refer to an individual plate. The most usual way, most common, perhaps the most practical way of doing that is one that we earned from, we received from an American investigator named Charles Kofoid, and it's been in operation since the early 20th century, and it divides the plates in equatorial rows, starting with the apical row, going to the one just before the cingulum, which would be the precingular row, then the singular, then the postsingular, and then the plates on the antapex. And then, of course, they are numbered in each row in the normal way that we write from left to right, which gives this result that is here. When there are rows between the plates, we just call them intercalary and add a letter instead of a prime as a code for these plates. So this is the Kofoidian system that is used, and it's very useful to refer to these things. We have within the Peridinialis, most commonly the genus Peridinium. You can see here the tabulation typical of the type species, Peridinium cinctum, with this pointed plate that is pointed to the right side of the cell on this side. So an asymmetrical tabulation that is characteristic of the species, together with this notch here and the epitheca. And another common species, or at least common in some parts of the world, I have to say, if Cowley is there, we found it. We found it in Alentejo. One single puddle, one single lake, about 25 meters across, was completely full of it. We never saw it anywhere else in Portugal. So these things happen. And here, this group of species, they all look like peridinioids at first glance, but the upper two have actually been transferred to the Pterocosphoralis. This is actually the Tyrannodinium, the fusariation that we saw feeding before. This is, it used to be Peridinium lomnici. It is now Chimonodinium lomnici, close to the Scriptiella group, close to the basis of the fusariations. This is Palatinus. This is now in a separate family within the Peridinialis, Peridiniopsidaceae, and this is actually not within the Peridinialis or Pterocosphoralis at all. It is within the Suecialis. It is what I call Spherodinium cracoviens. So I don't want to detain you any longer. I have already passed the time. So that's what I would like to say.
Thank you.
I hope that the choice of subjects was useful for you. Without previous information, I had to choose which way to go.
Alguma pergunta, pessoal, a mais? Eu tenho uma pergunta. Dentro da sua visão, com relação às diferenças dos dinos marinhos de água doce, e assim, tendo em vista que é um grupo que eu costumo brincar com os meus alunos, assim, que Deus, quando estava fazendo as algas, ele quis brincar e falou assim, ó, vamos escolher um grupo para a gente fazer experimento, né? A gente brincar um pouco. Aí um dos grupos que ele escolheu foram os dinoflagelados, né? Que é tudo de exceção, muita coisa diferente aparece nos dinoflagelados. Então, tendo em vista essa diferença do marinho e de água doce, e ao mesmo tempo essa capacidade tão grande, essa maleabilidade tão grande de diversidade, o que se pode esperar para a questão de mudanças climáticas, por exemplo, os efeitos sobre esse tipo de organismo? Porque a gente verifica muito disso para cianobactérias, que são extremamente resistentes, né? Mas os próprios dinos, eles são muito dinâmicos. Então, o que você acha a respeito, assim?
Eu julgo que poucos deles, a não ser os que vivem em habitats que vão estar eles próprios ameaçados, por exemplo, espécies que vivam em alta montanha, eles estão em maus lençóis. Espécies marinhas que vão apenas modificar a sua gama de ocorrência e provavelmente, se juntarmos a isto a poluição costeira, vamos ter espécies a fazer mais volumes. Temperaturas maiores, zonas adequadas com nutrientes suficientes e vamos provavelmente ter um agravamento dessas situações. Portanto, poucas espécies provavelmente vão estar em situação de desaparecer, vão expandir algumas delas, alguns dos seus limites geográficos. Eu só vejo mesmo aquelas que já estão perto dos limites, como por exemplo espécies de águas limpas, águas pouco mineralizadas de alta montanha, que precisam de águas frias e que começam cada vez menos a ter águas frias e não têm ponto de fugir. Que aliás é o mesmo que está a acontecer com plantas alpinas. Já não têm como sair dali, não é? Não têm como sobreviver em zonas mais baixas para conseguir colonizar outras áreas e estão a ser empurradas até ao limite e muito provavelmente estão em maus lençóis. Mas muito do que possa ocorrer de facto, eu diria que podemos e devemos tentar prever para cada caso. Mas também diria quase certeza que vai acontecer outra coisa.
Uma pergunta da Maíra aí.
Quais os géneros produtores de toxina em água doce? Só está assinalado costumava ser chamado peridínio polónico ou peridiniopsis polónica, e agora há um género diferente para ele, que é o género Naiadinium. Naiadinium polónico foi descrito como tendo produzido uma toxina nos lagos no Japão, e eles chegaram até ao ponto de caracterizar quimicamente a toxina, que chamaram, à base do nome polónico, polonotoxino, qualquer coisa com polónico. Portanto, há uma caracterização, há esses registros, portanto, pelo menos dos anos 60, 1965, penso eu, do Japão, depois talvez um outro também do Japão, e se bem me lembro, em Espanha, onde não caracterizaram a toxina, mas descrevem a ocorrência de mortalidade de peixe coincidente com a ocorrência desta espécie. Eu já a vi várias vezes, às vezes até em quantidades grandes. Tentei saber se alguém tinha visto alguma evidência de mortalidade de peixes ou alguma perturbação do ambiente. Nunca me deparei com essa situação. Portanto, está referenciado, houve caracterização de um composto, mas parece ser, de facto, uma preocupação menor, pelo menos até ver.
No caso, os dinoflagelados, se aplicaria também aquele conceito que a gente utiliza para cianobactérias, de potencialmente tóxicos? Ou
eu diria que em água doce, como digo, os casos documentados são tão poucos e referem-se à mesma espécie que até haver mais evidências e mais casos eu não iria tão longe até ao ponto de chamar potencialmente tóxicos. Enquanto que nas cianobactérias, sobretudo quando elas ocorrem em blooms, a probabilidade de ter algumas que sejam de facto produtores de toxinas é, eu diria, bastante grande. Bastante grande. A quantidade de compostos produzidos por cianobactérias é enorme, não é?
Sim.
Aha. A question about the Varnoviids. Yes, Varnoviids. They are perhaps some of the most spectacular cells that exist in the eye or the ocelloid. Well, what is known about the ocelloid is that unlike the photoreceptors that you have, for instance, connected with the flagella movement and so forth, which can detect intensities in certain wavelengths, the ocelloid actually has a lens, and apparently it can actually make an image. So this is a very complex structure, and it is really complex. We've been trying to reconstruct in 3D one of these structures, and we have currently in the lab a set of a few hundreds of sections, of photographs of sections to reconstruct these structures. They are incredibly complicated, so obviously the cell uses it. How it uses it, well, I think it's still anybody's guess about the mechanism. But the fact is that the cells that have the ocelloids, almost all of them, have at the same time the so-called nematocysts. And the nematocysts are these sort of harpoons that can be shot into a prey. So I would think that there will be a connection between the two things. So the ocelloid with its lens that can form an image, perhaps it can help the cell distinguish between a prey or something to which it would be useful to send an harpoon from something else that is not worth spending this very complicated machine on. The harpoon itself, the nematocyst, is also incredibly complicated. So I can see the reason for trying not to waste this ammunition in items that are not worth it. So, but how it works? Well, I expect that if you solve that, you'll have a Nobel Prize waiting for you.
Nós temos outra pergunta sobre o bendito Ceratium.
Mm-hmm. Well, the first question is it really the same? What you've been having is everybody was calling Serratium rudinella to any freshwater Serratium with 3 horns or sometimes one apical and 3 antapical, and that went on for quite a long time until it became clear that they actually have different tabulations. So we have basically you have 2 described species, Rudinella and Fucoides, and they may behave differently in terms of ecology and in terms of how they handle nutrients. One thing we know, they can co-occur. We've seen lakes that were quite full of serratium, and you had the 2 species side by side. You could have them in the microscope in the same slide. I actually have published a photograph that is not. It was not mounted. They are side by side because they were like that in the microscope. So they can co-occur, but this doesn't mean that the limits are the same. So the first question is whether you have the same. The other question, of course, is whether there will be more species, because these are known to have quite some variation. One species that has been described is a smaller species with shorter horns that was described with the name Serratium rhomboides. Is it a real species? Or are we just looking at some morphological variations of a continuum that could contain a lot of cryptic species? So I think only after starting to really analyze the genetic variation in these things, then we may start to understand whether we are really looking at a versatile species or whether we are looking at actually at several different things. This is it is possible, but it is demanding, because if you want to relate morphologies, especially aspects of morphology which you don't know if they are significant in terms of defining different things, I only see one way, is to pick up single cells. You document single cells, photograph them, film them, and then pick them up, PCR, amplification, and if it's successful, you can connect a sequence to an image. And this, of course, will not be 100% successful. So it means if you want to do this, you really want to do a lot of it, a lot of isolations, a lot of attempts for PCR. But then you can find whether, if you choose your markers right, if you're lucky, you can find whether they all fall within the same group or whether you have several clusters. And if you have several clusters, you can look at the images again and see, why are these different? Because a priori, you don't know which, you see variation, which differences are meaningful in terms of genetic variation. I don't know. I need to see the genetic groupings, and then I go back and see if there is something that will be meaningful in terms of variation. That's the only way that I see that one can start to disentangle these things.
Yeah. Okay. Any other questions, people? Okay. António, I would like to thank you so much. It was a pleasure to receive you here. It's an honor for us also, because we are trying to get some classes with the specialists around the world, and your presence here talking about dinoflagelates. It's very, very important for us. Thank you so much. And the last time that we have, if you want to say something.
Well, just I hope that you have benefited from the choice that I made. I was not sure whether to go more into this direction of presenting the group, or if you would like more to go to the more practical classification of freshwater dinoflagellates. So it was a choice. I just hope that you have enjoyed it, and there is some benefit from it.
Yeah, of course. The lecture was very, very, very good. And we have also a lot of comments, elogios, and congrats for your lecture, your class also. Then after we can read that in the comments of the YouTube video, ok? Obrigado também. Então, pessoal, à tarde nós teremos Periphyton com a professora Bárbara Dunque e a professora Denise Bicudo, ambas tratando sobre a temática Periphyton, né? E duas vertentes diferentes. Bárbara falando um pouco sobre a questão funcional, Denise falando sobre a estrutura em geral. Então, aguardo vocês às 14 horas, ok? Então, até mais, pessoal. Obrigado.